snapshots. And before going on, I'm going to tell you one of those fundamental limits because it will play a big role in the second half of the talk. But the bottom line is that this is what motivated us to transition to an experimental approach. So here is one of the limits that we found. So imagine you have a single cell snapshot, and then let's say that the reality of the situation is that cells in a given state are not all moving in exactly the same way. So for example, in a particular gene expression state, some subset of the cells are upregulating a certain gene, and some set of them might be downregulating a certain gene. There are two scenarios that might explain this phenomenon. One is that it's just a purely stochastic thing. So gene expression is really noisy. Even cells that are trying to maintain the same state over time, sometimes a gene will fluctuate up, sometimes it will fluctuate down. And for that reason, if you look at any instantaneous moment in time, some of the cells might be increasing the gene and some of them might be decreasing the gene. Another possibility is that the cells, unbeknownst to you, there's some sort of variable that you can't see that's not in the single cell RNA-seq measurement, but that actually distinguishes the cells into two different groups where, and for example, that would be colored as red and blue here, and where one of the groups is consistently upregulating the gene and another is consistently downregulating the gene. So an example might be that their cells have some sort of epigenetic state. And the epigenetic state, like whatever chromatin marks and stuff like that, biases them toward one fate or another fate. So even though the cells by single cell RNA-seq might all appear to be in the same progenitor state, some of them are committed and consistently going to go in one direction and others in another direction. And because by the definition of these states is that they're hidden, therefore they represent a fundamental limit on the accuracy of our predictions when we try to infer cell fate from snapshots of single-cell RNA-seq data. So just remember that, because we will return to it. And for the people who care or appreciate the language, the existence of these hidden variables or hidden states would cause the process to be non-Markovian with respect to the things that we can measure. So another way to phrase the fact that we have to assume there are no hidden states is that we can do no better than roughly to assume that the process is Markovian with respect to what we can measure. Okay, moving on. So what's the experimental approach? So ideally what we would like to do with an experiment is measure the state of a cell and then look at what it does later. But that, as we've now discussed many times, is not currently possible because we kill the state in the process of measuring it. So an alternative would be to let a cell divide, then measure one of the sisters, and then let the other sister differentiate, and then look at what it did. And as long as the two sister cells were similar to each other, right at the moment when we measured the first one, then we have a kind of approximate version of a map between the state of the cells and what they did later. At this point, I would like to pause and thank Ale, who's the co-star of the second half of my talk, and a really amazing collaborator, and also Fernando, his advisor, for working with us on this project. It was a real privilege. So, and the thing that, among many other things, the initial tool that Ale contributed to this project was a barcoding lentivirus that allows us to track clones over time. So the way that this works is that we use this lentivirus to genetically engineer cells to express a GFP gene, so they turn green. And then the main point is that in the mRNA of that GFP gene, there's a random barcode sequence. And when the cell divides, the two daughter cells both get that barcode, and we can read the barcode when we do single-cell RNA-seq. So this gives us a way to track over time which cells are descended from a common ancestor. So the idea of how this experiment would work is that we would take a group of cells, and these cells would be like cells that we had just taken out of a mouse's bone marrow, like hematopoietic progenitors. Then we would use a barcode to label them, then let them divide for 2 days, then split them randomly into two groups, and by chance, like these two red cells descended from this red cell, you'll get two sister cells that end up in different buckets. Then one of the buckets we just measure immediately, and then the other one we could let it differentiate in a culture dish or back in a mouse, and then look at what it did later. And then use the barcodes to associate the sister cells, and then see how the state of the cell correlates with what it did later. So as everyone is familiar with, experiments don't usually go like you plan at the beginning. And so for me, being used to the kind of quick turnover of computational work, it was really depressing to start doing experiments. And it definitely took a toll on my mental health. And I don't know if you guys have been to the Klein lab, but there's rubber duckies everywhere. And also I used to have a razor on my desk, and it was in this time that I started mutilating rubber ducks. Okay. So, anyway, rubber ducks aside, the experiment did eventually work, and thus this was the end of this collection. And here I'm showing you data from the in vitro version of this experiment. So, okay, so what's going on? So basically, in this experiment, I did what I just described, harvested hematopoietic progenitor cells from a mouse, then barcoded them, then then after 2 days of them dividing, I took half the cells and measured them, and they're being shown. In this bottom part, gray is all the cells, and the colored dots in each figure is like the cells from that time point. So I measured some of the cells after day 2, then the rest I let differentiate, then looked at some more cells, and then the rest of them differentiated. So this is an example of what the data looked like. Each of these figures is showing a different clone. As before, gray represents all the cells in the data set, and the colored dots represent the cells that belong to that clone. And so, and the color of the dot is what day it came from. So you can see like 3 different behaviors being shown here. Here, this is a clone that is just going straight to being a neutrophil. Then this one is differentiating and then bifurcating into 2 different fates over the course of 6 days. Whereas this clone seems to be like staying roughly where it is, so it's kind of self-renewing in the same state. So the goal is to move from these kind of anecdotal examples to get a systematic picture of how cells moved over the 4 days of the experiment. And as I mentioned before, the key requirement for us to have an accurate picture of this map is that the 2 sister cells be roughly similar at the time when I measured the first one. Otherwise, it's like not a good approximation of what the cells were doing. So what I'm showing in this figure is evidence that the cells were in similar states. Sister cells started out in similar states. So here, this is like all the cells, and the colored dots are clones from day 2, and each color is a different clone. So for example, these green dots are all descended from the same ancestor cell from 2 days earlier, and as you can see, they're roughly all in the same state, and same for the other clones. And then to look at this systematically, we took the day 2 space and like set of cell states and clustered them, and then what's being shown here is like a heat map, which is the joint distribution of how pairs of sister cells were in those different clusters. So the intensity of the IJ entry of this matrix is how many sister cell pairs were where one was in cluster I and one was in cluster J. And the point is that the fact that there's dark on the diagonal is an indication of the fact that they were usually in the same cluster. All right, so now we're prepared to get the picture of how the cells changed over time. And that's what's being shown here. So in this figure, each colored outline is one mature lineage. And the colored dots are cells from day 2, which are being colored by the dominant lineage attained by their sisters at a later time point. So in other words, a blue dot here is a cell from day 2, which had, where most of its sister cells on day 4 and 6 ended up as a monocyte, and same for green, et cetera, et cetera. So what you can see is that as we get closer to the mature fates, then the cells that are like on their way into that fate are almost exclusively become like fated for that lineage. But then in the middle, then there's a mixture of cells that have the potential for going to different fates. Thanks to the ALE, we also were able to do this experiment in vivo. So this is showing a similar experimental design, but now instead of culturing the cells, we transplanted them into a bunch of mice and then harvested them a week or 2 later. And so what's being shown in these figures is the day 2 cell landscape where the color of each cell, so in this figure, yellow means that the cell became a progenitor cell, like it basically self-renewed inside the mouse. Whereas in this figure, yellow means that the cell its sister became an erythroid cell inside the mouse. So basically the fact that yellow is in different parts of this plot means we can see how different parts of the day 2 state space are faded for different lineages inside the mouse. So now let's return to our whole goal in doing this experiment. So the issue that with potentially the issue with using facts as a way to define the cell state landscape is that the cell states weren't pure. So for one thing, they weren't pure in their gene expression state, but also they weren't functionally pure. They contained mixtures of cells with different properties that were committed to different lineages. So for single-cell RNA-seq to be a good revision or a good add-on to this, we want single-cell RNA-seq states to finally be pure. And otherwise, we have to keep adding new measurements to get a space that truly represents what the cells can do. And as you can see here, there are parts of this space where cells seem to be fated to do different things. So we may here be back in the problematic situation of facts, where even though a cell state even though there's only one cell state here, from the perspective of single-cell RNA-seq, it actually contains cells with a mixture of different properties, and thus there may be additional things we need to measure to truly get a state space in which each region is functionally pure. And this goes back to the fundamental limit of hidden variables, because the existence, the fact that cells with different functions would be in the same measured gene expression state would imply that there are hidden variables. So, but we may actually, this salt and pepper thing may not be just strictly a problem with single-cell RNA-seq as a measurement. It might just be because of undersampling. So, a cell we may only observe it to make a monocyte because we only sampled one cell from 4 days later, and that cell happened to be a monocyte. So then it would seem like that cell was 100% going to make a monocyte. But if we could get this cell to be in infinity copies and sample all those copies, we would get the true distribution. So just because of sampling, we might see different cell states appearing, different fate potentials appearing to coexist in the same state. So we have to figure out some way to overcome this. And I will now describe to you the test that we use to figure out if there's hidden variables. So to think about this analysis, we're going to play an imaginary game. So in this game, we take a progenitor cell and let it divide. And one of the sister cells I'm going to give to you, and one of them I'm going to keep for myself. I'm going to take my cell and put it into a culture dish in my lab and let it differentiate and then see what it turns into. You have to now predict what my cell did. Okay, so you have 2 options for how to play the game on your end. You can either look at the cell immediately with single-cell RNA-seq, or you can take your cell, put it into an identical culture dish in your lab, and then look at what your cell did after the same amount of time. And you also have access to 1 million previous examples of this game. So I claim that if there are no hidden variables, it's always better to look immediately. But if there are hidden variables, it might be better to look later on. The reason is, if there's no hidden variables, then everything there is to know about the cell is in the stuff that you can measure right now. And by putting it into a culture dish in your lab, the only thing that can happen is to introduce noise and to corrupt your ability to predict what's going to happen. On the other hand, if there are hidden variables, then by looking now, you may have lost an opportunity to let the cell bring those hidden variables to the surface. Say it's a chromatin state, the chromatin state may eventually manifest itself in directing the fate of the cell. So this can also be framed in a more rigorous way mathematically. So the idea is that if there's no hidden variables, then the 4 cell states, sister 1, sister 2, and then the fate of sister 2 and the fate of sister 1, those can be thought of as random variables. And if there's no hidden variables, then these 4 random variables form a Markov random field with respect to this graph, meaning that any 2 nodes are conditionally independent given the nodes that lie between them. And in this situation, there's something called the data processing inequality, which says that anytime you have a kind of Markovian process like this, then let's say like this node is more predictive of this node than it is of that node. Like you always lose information by moving through the diagram. So in particular, like quote unquote what we're calling forward prediction, the prediction of D from A will always be worse or equal to or worse than the prediction, what we're calling lateral prediction, the prediction of D from B. So, so the bottom line is that if lateral prediction is better, aka if there's a violation of the data processing inequality, that means that this thing is not Markovian and therefore there are hidden variables. So if this is better, then there are hidden variables. That's the bottom line with this slide. So in the in vitro experiment, we got an opportunity to actually compare directly the accuracy of these, the fidelity of these relationships. So as I mentioned before, we have a bunch of examples where we have a cell, and then we measure the fate of its sister cell, and that enables us to do a measure of forward prediction. There also, because we cultured the cells in two different, like at the beginning of the experiment, we split the cells. They're the ones we looked at right away, and then the ones we didn't look at, we put them into two separate wells and let them differentiate in parallel in separate wells. And by comparing the ability to predict what happened in one well from what happened in the other well, that gives us an example of lateral prediction. So, so therefore we can directly compare lateral prediction to forward prediction. So here I'm showing you, okay, so this figure gives us a sense that lateral prediction might actually be pretty good. And the idea is that in this heat map, okay, so there's two heat maps. Each one corresponds to one well. Each row of the heat map is a clone, and each column is a different lineage. And so this is showing the distribution of lineages that occurred in each well. Basically, the fact that these heat maps look similar to each other means that roughly the same fates occurred in both wells when it was the same clone. So basically, lateral things usually did the same thing in these two wells. Bottom line, when we use a bunch of different machine learning methods to try to predict, to do lateral prediction versus forward prediction, what we consistently see is that lateral prediction is a better predictor than forward prediction, meaning that in the system there do appear to be hidden variables. So there's things that the cell doesn't reveal on day 2 that, that nevertheless manifest themselves in the function of the cell. And so this is basically bad news for this particular, or it's not bad news, it just means that the march goes on for more accurate measurements. And also there could be several different phenomena that cause this. One is that, so the hidden variable may consist in like protein in the cell, the chromatin state of the cell. It might just be in transcripts that we weren't sampling because single cell RNA-seq is a very sparse measurement. So it's possible that if we could just do a deeper measurement of cell state, then these like points might converge back to the line. And interestingly, this is also true in vivo. So in the in vivo experiment, we kind of have a similar situation where we took cells and we transplanted them into, for the time point that we're looking at here, 6 different mice. And so now the question is, how well can we predict what happened in one mouse from what happened in the other mouse? So this is like a similar situation where these two heat maps represent 2 different mice. Now because there's 6 mice, it's just each row here is like represents 2 different mice, but there's different pairs of mice on each row. But the point is that it's functionally the same type of figure. And so we can make the same comparison. How well can we predict this side from this side compared to the forward prediction? And again, lateral prediction is better. So that shows that even in vivo, where the environment is a lot more unpredictable, and you might expect that cells wouldn't be so consistent in what they did, even then, it's still, like, you still get a better prediction by taking your cell and transplanting it into your own mouse and seeing what it did in your own mouse. Okay, so that's the end of that. Actually, that's the end of the entire talk. And so what did we learn and why did I call it a story of limits? So first of all, there were limits on computational inference. That was the big final message of the part 1 of the talk, which is like that since we can't asymptotically predict what the cells are going to do, no one can. In fact, there's fundamental limits on the ability to predict cell fate from snapshots of single-cell RNA-seq. The second type of limit that was interesting to see is limits on information about fate choice contained in at least the existing measurements of single-cell RNA-seq that we have. So even like setting aside what the limits on what we can infer just without extra data, even if we had like a perfect measurement of what cells did, we would still not be able to fully like predict a cell because cells with different functional properties can be superimposed in roughly the same gene expression state. At least gene expression as to the extent that we can measure it with the existing tools. Finally, there are limits on math and limits on like what I can sort of on the initial ambition that I had to do stuff with math. So I sort of came into this department being all high on pure math and being excited to like solve problems with math. And I think what I learned over time is that math has a certain role, but it's not like you shouldn't have like a math savior complex in the approach to problems. And actually for me, the function of math ended up being in kind of framing a certain problem and indicating which experiments would be good to do. And I think in this sense, it was a very classic systems biology experience, because this is a department which is all about the kind of fusion of math and experiments that actually have like an ongoing conversation as opposed to being like encamped in their separate territories. So I feel really lucky to have gotten to be here and to have had that experience. So, okay, that's the end of the science, and now I get to thank people. Okay, well, first, I didn't make a slide for this, but I just want to thank everyone in this room for coming, and you guys are all awesome, and there are people, well, okay, some have slides, but I have friends from high school here, people in my lab, people not in my lab, people that I've known from all walks of life, and I just really appreciate it. So, great job for coming. So, okay, next I have very specific thank yous for the admin folks who really worked with me specifically with the Klein lab, and they just made everything really seamless, and I'm really grateful for them. Then my lab managers, who I guess this is sort of a cliche, but it's like you know how amazing their work is when you don't even know that it's going on. Except I do know it's going on, and I really appreciate it. And also the single cell core, so that these are the group of people who kind of maintain all the materials and reagents that we need for doing single cell RNA-seq experiments. Also the members of my DAC and my thesis committee. And also the people I had the opportunity to work with over the years on the diversity and inclusion projects. And she's not here, but especially Daphne has been an amazing collaborator, and I'm really lucky that I got to work with her. And okay, next I'd like to thank Ale. So Ale is basically the hero of my PhD, and he just made everything possible. And like every time I'm like, oh hey Ale, like wouldn't it be cool to do this experiment? He's like, yeah, I'll do it. And I'm like, what? Wait, oh whoa, okay, fine. And so yeah, so Ali's really talented, and he's a great experimentalist and also a great thinker about biology, and also just a very gracious and good guy. Like a story recently happened, I'll tell you a story of something recently happened. So basically, I was in a meeting with Alon and Ale, it was over like Skype or something, and we had just gotten reviews back from a paper. And so we were talking and, you know, deciding what to do next, and at the end, Ale was like, I just want to say it's such a privilege to work with you guys. Like I really love that I get to work with you guys. And then we were like, oh, thanks, like, okay. And then we hung up, and then Alana and I were like, what, like, who does that? And the ironic thing is that we're the lucky ones to work with Ale. So it was honestly just an absurd thing for a person to do in that situation. But, you know, Ale is just absurdly gracious type of guy. Okay, next I would like to thank my girlfriend, Julia. So we have been dating for 8 years, and we live together, and obviously there's a lot to say about someone in that situation. So, you know, but I have to limit my remarks here. And therefore, I'll just try to concisely frame the situation as that she's my hero, and I just want to be like Julia when I grow up. And everything I do is like a imperfect attempt to imitate Julia. And So for example, Alon mentioned that I do this like workshop about diversity and inclusion, but it may seem like I sort of came up with that. I'm like, where did this guy get it? But actually, I'm just copying Julia, because Julia runs a similar workshop teaching Jews about racial justice. And if any of you belong to a synagogue or a Jewish community, then you can just email me, because it's a great workshop, and we're looking for clients. And so another example is that Julia is a feminist, and I just want to be like her and be a feminist. So yeah, I'm excited and to just keep, and also she keeps changing and improving, so I never quite get there, so I just get to be led along with like this carrot on a stick of like the awesomest way to be. So I don't know how Julia got so awesome, but you can partly lay the blame on her family. And so this is the Stoller-Lieberman-Korn family, and again, there's like a lot to say about an entire complex human being, and especially 6 complex human beings. But the bottom line is that what I especially treasure about Julia's family is how much they value family. And so the fact that they flew here from Chicago to come to this talk is evidence of that. So I really appreciate you guys for coming. Okay, next I'd like to thank my family. So they're my crew, and they live in Newton, so I see them all the time. And yeah, I mean, I'm facing the same problem that's been a recurring theme here, which is like, what do you even say about people that you're so close to? But I'd especially like to give a shout out to my mom. Also, my dad couldn't make it, so I don't have to worry about this. So basically, well, 2 things I'd like to say about my mom. First of all, she can get some of the credit for she's also a social justice warrior. And I should mention that she also teaches a class about diversity and inclusion and identity. So I'm also sort of copying her. And but also, it was in my upbringing that she can be credited. So the situation is that in my basement, I used to do art. And I would so throughout my childhood, the following scenario would play out on like a monthly basis. I would like be in the basement, like cranking away on whatever. Every time I was done, I would like put a box or like drape or close the door so that my mom wouldn't see it. And then at the moment of its completion, I would bring it up to my mom and be like, check it out. And then invariably, no matter how good or bad it was, she would be like, oh my God, like, you might have to hang this in the Louvre. No, don't, I want to keep it in my house. So yeah, it was like a really weird level of freak out. But I think it served me well, because now, even as I've left the house and I don't do much art anymore, I'm like continually in search of the affirmative freak out. And so like my whole life is just like a desire to get people to react the way my mom did, but in a good way, where it's like, yeah, so basically I think that's why I'm always like excited to work for like positive affirmation, and she set that pattern in motion. Okay, next I'd like to thank Alon. So Alon is a really great guy. He's also a big mensch, and he's almost, like from the outside at least, annoyingly perfect. Like there's always if there was like a movie about Alon, it will be a really boring movie, at least for but basically the movie would be like a montage. It would be like Alon walking with his daughter, like cut. Then Alon is like in the office, like really thinking about someone's problem, like that's the only thing he cares about. Then like next cut, like Alon having fun in a fun situation. Next cut, Alon being serious when it's demanded. And just, whatever the situation, Alon's just like perfect, always has the most apt right behavior for that situation. And I'm always like writing, I'll like try to send an email to someone or whatever, and I run it by Alon. I was like, no. This email has to be sent this way. I'm like, where do you learn all this stuff? But anyway, I'll just say one of the features about Alon that I've really benefited from during my advising, well, to explain this, I have to make an analogy to biology. So in biology, in biological systems, there's often a trade-off between robustness and sensitivity. So, for example, basically, biological systems want to be robust. They don't want to be just wildly changing in response to perturbations that they get from the environment, but often they need to be very sensitive to the perturbations that matter. So, for example, T cells, they don't want to freak out and start, developing T-cell sensitivity to every antigen that comes their way, but when that one virus or bacteria or whatever comes their way, then even like one molecule of it, and they need to react. And so that's very difficult to achieve simultaneously robustness and sensitivity. So, but Elan manages, even in excess of T cells, to have the combination of robustness and sensitivity. Like, no matter what the situation is, Elan's like a very stable, reliable human being. Like, always, like, whatever's happening, he's always like, you can just go into his office and trust that he'll just talk to you, and his interests roughly don't change over time. And yet, when you bring like a new idea, then he's all over it and he really reacts. And so an example of that is when I came in with like different math ideas, Elan would like jump up and he's like, oh my god, like this is cool. Then he would like start going on the board. Like before long, Elan had read the math paper that I was citing and in more detail than I had. And another example is with diversity and inclusion. Like maybe you were into it before, but the point is that the point is like I went into your office, and at least once you knew that I cared about it, then every subsequent meeting would be like, oh hey Caleb, I just had these thoughts about diversity and inclusion. So I think that that's really awesome. It's hard to find someone who's both really stable but also really like reactive to whatever you bring into the room. Okay, finally, I'd like to thank my lab. And so first, I will explain the whole rubber ducky situation with my lab. So the rubber ducky theme has, because basically like now, I just want to set the record straight for the community. So it started when members of the lab, before I got there, played some sort of prank on Elan, I think, where they created like a whole bathtub and filled it with water in his office, and then adorned the bathtub with rubber duckies. And so then, after the bathtub had left, we just had rubber duckies sitting around the lab, and then we all just started throwing them at each other for some reason. And that was like a thing that just throughout the day, rubber duckies would come hurling into your vicinity. And so then, and at that point the lab was all male. So, which is relevant. And so then at one point, two new students, Haley and Kalki, joined the lab, and within the first like week or two, I remember one of them came up to me and was like, do we get rubber duckies? And then I was like doing a workshop on diversity and inclusion at the time, so I was like, it was like a 3-fire alert. I was like, oh my god, inclusion, they need rubber duckies. And, so I promptly ordered 100 rubber duckies on Amazon so we would never have this problem again. And but the Klein Lab is really cool because before long, rubber duckies just started appearing in random places. Like I don't know who did all this, but like for example, these ones are held there with magnets. And people also started bringing like novelty rubber ducks from different places that they went. And like, every time people would go on vacation, they would bring back like a rubber ducky themed of that vacation. So rubber duckies are now a big part of our lab life. But I also think it's apt that they're our mascot because the Klein Lab has a lot in common with rubber duckies. So for example, in the Klein Lab, people are really athletic and they're into going on races and stuff. And rubber duckies too are also known to go on really big races every year all over the world. Another similarity is that rubber duckies have contributed a lot to science. So there's actually a famous case of a ship capsizing, and 30,000 rubber duckies ended up in the water. And over time, they were observed washing up on the shores all over the world. And this data has actually been meticulously tracked and used to trace ocean currents. Rubber duckies have changed a lot over time. So this is from 1949, the patent for the original rubber ducky. Here's a sample of rubber duckies in 2019. And in a similar way, the Klein Lab has gotten a lot more diverse over time. Finally, the main similarity that the Klein Lab has with rubber duckies is their fundamental goodness. Like staring into the face of this rubber ducky, it's all just innocence and love, and you just want to be its best friend, and it's just so like generous and awesome, and that's essentially the situation with the Klein Lab. So yeah, I just feel really lucky to have had that rubber ducky, namely the Klein Lab, as my friend, and okay, that's the end of my... So I think now I'm supposed to answer questions.